Hey guys, welcome back to the channel. In this video, I'm going to be covering IB Biology 1.3 in under 10 minutes. So it's going to be the third video of my under 10 minutes topic series. So the first thing you need to know about 1.3 is that the cell membrane is not just this random border around cells, but it's actually this intricate structure. And for this intricate structure, as you can guess in science, we've had different models that represented it. And these models have changed over time. So the first model we had was this thing called the Davson-Danielli model of the cell membrane, and what that described was it was basically the phospholipid bilayer on the inside, and around it were membrane proteins. So the reason this was proposed was because when you looked at electron microscopes, you had the cell membrane sort of looking like two dark lines with a sort of light bit in the middle, and phospholipids appear lighter, whereas proteins appear darker on under electron microscopes. So people deduced that the scientists deduced that these were proteins and these were phospholipids, right? So that was the reasoning behind the Davson-Danielli model. But as you can guess, we've moved on to a different one called the fluid mosaic model, and this is called the Singer-Nicholson model. Now, the Singer-Nicholson model has many features, such as membranes that go through the proteins that go through the cell membrane, as well as different molecules that are within the cell membrane as well, such as cholesterol. So the previous version did not have this, but this version did. And the main techniques that proved this were called fluorescent antibody tagging. So fluorescent antibody tagging. This just involved giving membrane proteins different colored antibodies so that they'd be different stains. And after a while, they found that these colors would mix, suggesting that these membrane proteins were able to move laterally across the cell membrane, suggesting that it's fluid, going against the Davson-Danielli. And another technique they used was the analysis of membrane proteins. So what they found was membrane proteins actually had regions of hydrophilic and hydrophobic regions, so it would be unlikely that they would just be a layer surrounding the phospholipids, but rather they'd go through. Another technique they did was called freeze fracturing, which involved freezing cell membrane and taking a picture. And what they found was there were sort of like these cracks in the electron micrograph, and these were going according to membrane proteins, suggesting that membrane proteins go through through the plasma membrane, which is what we use now. So this is what disproved the Davson-Danielli model, and we have the Singer-Nicholson model. So the Singer-Nicholson model is called the fluid mosaic model, and this is what it typically looks like. So these things right here are called phospholipids. A phospholipid is a molecule which contains a phosphate head and two fatty acid tails. So these fatty acid tails are going to be just lipids without any particular charge. So there's going to be hydrocarbons. So this is going to be hydrophobic, and this is going to be hydrophilic. So water, yes, no water, no. All right. Kind of looks like weird water, but that's basically it. That's why an emergent property of these phosphates, a property that works from multiple things working together, is that when you put lots of phospholipids in, say, a container, what happens is these things arrange themselves such that the hydrophobic regions are sort of facing inwards, and the hydrophilic regions are facing outwards. Sort of like a defense mechanism, essentially, because they're bits that don't like water face inside and don't face the water, and bits on the outside that do actually face it. So the Davson-Danielli the Singer-Nicholson fluid membrane is, or just fluid plasma membranes in general, are 10 nanometers thick. And what we have, the first kind of protein is called the integral protein. But this one is a peripheral protein, sorry. So a peripheral protein, as you can guess by the name, is something in the periphery. So it's only on one side of the membrane. It doesn't cross all the way through. And these ones we call them monotopic, and they're also temporary. So these things don't last forever in the membrane. Now the other kind are these ones called integral proteins. And when you think of integral, you can think of like integrals in maths, but I think of them as being important. And this is because they are permanently associated with the membrane, and they go all the way through. So they're called I think it's pluritopic. Yeah, that might be the word. And also another type of protein you might need to know are these proteins called glycoproteins. And what they are is just a protein with the glyco part, which means glucose. So you have these chains of polysaccharides, right? It doesn't necessarily have to be glucose. When you see glyco, you can just think of sugar. So they have these large chains of sugars that sort of branch out, and these are called the oligosaccharide. Actually, that's the specific name. And you'll need to know how to draw these structures because it's an essential skill in 1.3. Another important molecule, probably the most important one here, is cholesterol. Now, this is very applicable to our own lives because we always hear people having too much cholesterol, too little cholesterol, and foods that are high in cholesterol. And cholesterol, as you can guess in anything in biology, is not bad in low quantities, but can be bad when there's excessive amounts. So what cholesterol does is these phosphate heads are actually quite weakly associated, right? And they also they're not very tightly packed. What cholesterol does is actually it sort of acts like a glue, sticking these things closer together. But at the same time, it's sort of like a disruptive molecule that it doesn't fit this plasma membrane. So what it does, it makes it more fluid and able to move around, right? Along with that, what cholesterol does is it actually cholesterol in itself it's a fat, so it's associated with fat, so it is also non-polar and hydrophobic, right? So when molecules want to cross this phospholipid bilayer, cholesterol will actually help in stopping these polar molecules from crossing. So you know about endocytosis, exocytosis, and just stuff in a cell having to come in and go out, like oxygen and carbon dioxide. Well, that not everything can come in. So we call that the selective permeability of the cell membrane. And basically, what that means is some things can come in, some things some things can't come in. And usually, the things that come in are non-polar molecules that are very small. So non-polar, we draw this in green. Non-polar and small. So those are the ideal molecules that can come in, right? But what happens is it can sometimes very small polar molecules can come through. So cholesterol acts as a mediator because you don't want stuff going in and out of cells all the time. So you need to be able to contain the material. So it's good that cholesterol is there. But when you have too much cholesterol, you get other health consequences. But other than that, that's basically topic 1.3 for IB biology. Just know how to draw these structures and make sure some of the key parts are these phospholipids are there, and then try to make them be equal on both sides, and have at least three different proteins, membrane proteins, and sometimes people have an integral protein that is a channel protein, so it has like a hole that allows molecules to pass through, if you will. But I don't really put that. I put the integral protein, and I find it fine. And make sure you label the structure and label the phosphates. Make sure you label everything when you're doing a diagram on IB biology, because that's how you get the marks. And other than that, guys, that's my video for today. I hope you guys enjoyed. Make sure you like and subscribe. Leave a comment on any topic you want me to cover, because I'll probably go through these topics sequentially. So if you have a test coming up and you want me to cover a topic quite soon, then let me know and I'll cover it straight away. Alright, guys, have a good one.